Hello, good morning, and welcome to all of you. This is Rajesh Kumar Singhal, and I am here to present a new lecture on the light stress. So, friends, today we will discuss the different facts about the light stress. We will discuss that how light is important for photosynthesis and the growth and development. Then we will discuss that different component of light like light intensity and the light quality affect the plant growth and development. Then we will discuss about the photo inhibition due to the highlight. We will discuss about some leaf and canopy acclimatization acclimatization to reduce the effect of highlight stress. Also, we will discuss that how plants acclimatize their leaf and canopy to increase the light use efficiency. So we will discuss about some acclimatization response to the light stress. So let's start our discussion. The what is light, what is highlight, and how this light is crucial for the plant growth and development. So friends, you know that sunlight is one of the important energy source on this planet. Okay, so we can say that sunlight is the one of the natural energy source for the plant and the other organisms. So this sunlight act as a energy source, means to providing the energy, and second function, it act as a signal for the regulation of different plant growth and the development. Okay, so the sunlight act for the energy source in the plant, and also it work as a signal for the regulation of various plant growth and the development. Okay, तो ये आपके दो इंपॉर्टेंट फंक्शंस हो गए सनलाइट का इन द प्लांट्स. Now we'll talk about the light response curve. Okay, so friend, this is a light response curve. So in this curve, at x-axis there is a PAR, that is known as photosynthetic active radiation. Okay, so this is the light intensity, mostly 400 to 700 nanometer, which is crucial for the photosynthetic process. Means this is the active light responsible for the photosynthesis on this planet. Okay, and y-axis there is net photosynthetic rate. So friends, there is a negative value. This represents that here there is no light, no light and no photosynthesis. So these conditions occur during the dark conditions. So this is also known as dark respiration, means there is no light intensity and no photosynthesis. So photosynthesis where you are below than 0, okay, or 0. So now come to the second point, that is known as light compensation point. So friend, during the morning time when sun light coming to the plant, okay, so there is a initiation of photosynthesis, okay, initiation of photosynthesis, okay. So this is known as, this is the light intensity at which the photosynthesis just started, okay. Next is this is known as light limitations. So for friends, you know that one law given by the F. Blackman in 1905, that is known as limiting factors. So photosynthesis process is regulated by the different factors, like it depends upon the CO2 concentration, it also depends upon the temperature, and so many factors which regulated the photosynthetic rate. And light is also one of the components which regulate the photosynthetic rates. So Blackman told that, or Blackman says that when any component is in minimum quantity or minimum number, that regulate the photosynthetic rate. Means अगर यहाँ पे CO2 कम है तो CO2 limits the photosynthetic rate. If temperature is a limiting factor, then temperature limits the photosynthetic rate. So in this condition, light is the limiting factors. So when there is an increase in the light intensity, there is also increase in the photosynthetic rate. So at this time, light intensity is directly proportional to the photosynthetic rate. Means as we increase the light intensity, there is an increase in the photosynthetic rate. Next is the maximum photosynthesis. So this is the point or light intensity where is the photosynthetic rate is maximum, known as maximum photosynthesis. Then there is a light saturation point. So friend, this is known as light saturation point. Means at this point, if you are increasing the light intensity, there is no increase in the photosynthetic rate, okay. Means all the chlorophyll present in the plant are in saturated condition, so there is no increase in the photosynthesis. After this, there is a known as photo inhibition. What is the photo inhibition? Means at high light intensity, there is a damage to the chlorophyll or chloroplast structure, okay, which reduce the photosynthetic rate, okay. So this is known as photo inhibition. So आपको क्लियर हो गया कि लाइट रेस्पॉन्स कर्व में क्या होते हैं, डार्क रेस्पिरेशन होता है, मींस ड्यूरिंग नाइट टाइम होता है, तो हियर देयर इज नो फोटोसिंथेसिस. देन लाइट कंपनसेशन पॉइंट होता है, यहाँ से आपका फोटोसिंथेसिस रेस्पिरेशन बराबर होता है. देन लाइट लिमिटेशन फैक्टर होता है, मींस इन प्रोपोर्शनल होता है. As you increase the light intensity, there is also increase in the photosynthetic rate. Then maximum photosynthetic rate means at some light intensity there is a maximum rate of photosynthesis. Then there is a saturation point, means आपके जितने भी क्लोरोफिल मॉलिक्यूल्स है वो completely saturated हो चुके हैं लाइट से, so there is no increase in the photosynthetic rate, even there is increase in the light. Then photo inhibition means at high light intensity there is a damage to the chloroplastic structure or complexes which reduce the photosynthetic rate known as photo inhibition. Okay, so friend, this is very clear. Now you know that excess light is the light absorbed by the plant and the algae. So friends, you know that algae are the photoautotropic organisms which are responsible for the maximum photosynthesis on this planet. Okay, so when excess light is the light which is absorbed by the plant and algae that exceed their photosynthetic capacity. Means the light which is more than requirement. This is known as excess light. Okay. So this excess light is mediated by the some photoreceptors and the pigments. Okay. So what is photoreceptors? We already discussed more than 5 to 6 lecture in the development that these photoreceptors are the protein molecules, and also these are small organic compounds which have important role in the absorption of the light. And this photoreceptor is made up of protein and the prosthetic group that is known as chromophores. So this chromophore work in the absorption of the light. And in plants there are number of photoreceptors such as phytochrams. These receptors respond to the red and far light, cryptochrome for the blue light, rhodopsins, xanthopsin, blood sensor, light, oxygen, voltage sensor that is known as love sensors. And in this phototropin, jet loop, ionochrome, and PSII. So these are the some love sensors in the plant. Then UVRAD. This is also one of the light receptors respond to the ultraviolet light. So in plant there are number of photoreceptors which respond to the different types of light and regulate the development process. Okay. So this is about the light. Now come to the next point about the sunlight spectrum. So friends, you know very well about the sunlight spectrum that there are sunlight is a electromagnetic waves. Okay, so sunlight work as a both waves and the photons. Okay, means they have both properties, wave and the particle properties. Okay, so this sunlight is made up of gamma rays, X rays, ultraviolet rays, then visual rays, then infrared rays for the radio waves. So when you go from gamma rays to the radio waves, there is an increase in the wavelength. Okay. So you know that about the energy is inversely proportional to the wavelength. This is the very well-known formula in the physics that the energy is inversely proportional to the wavelength. So when we go from the gamma rays to the radio waves, there is an increase in the wavelength and decrease in the energy. Okay. Now for us, only three types of rays, which is known as ultraviolet rays, visual rays, and the infrared waves are important for the plant growth and development. Okay, so these are the rays present on the Earth's surface, and you know that most of the ultraviolet rays absorbed by the ozone layer, but due to the global warming and the ozone layer depletion, there is more ozone, this ultraviolet are coming to the earth, and mostly ultraviolet A and B are present on the earth planet. Then visual lights like Vibgyor that is known as violet, indigo, blue, green, yellow, orange, and the red. So this have high energy and red have low. Okay, so these are the waves. Then infrared waves. So this is about the spectrum. So this spectrum have two components. One is known as quality and second one is the quantity. So quantity we can correlate with the intensity and quality with the colors or the types of different types of light, like green color we already discussed before. Okay, so these are the qualitative traits. Okay, then next, that sunlight, we can get full sunlight at the upper canopy level. Okay, also some plant are getting which are grown under the canopy, or some are shade plant, they are getting partial sunlight. Okay, so this is also one of the condition that we are receiving full sunlight or the half sunlight or partial sunlight. Okay, then light also have two component, that is the energy already discussed, that wave and the particles. So energy, this is total irradiance received on the leaf surface, measured in the watt per meter square, means how much energy is coming through the light. Then second point is the photon flux, means per second how many moles of quanta or photons are coming to the leaf surface. So these are also 2 points. So this photon flux is also known as photosynthetic photon flux density. Okay, flux density. So generally during the sunny days we are getting 2000 micromole per meter square per second. Okay, so this can be asked in the objectives, that how much we are there in the sunny days. Okay, next, sunlight can be the limited or low light stress, like in some northeast India you are getting the low light stress, and excess light mean you are receiving the high light. Okay, so friend, these are the component of the light, that different component, quality component, quantity component, full sunlight, half sunlight, energy, photons, or the high or the low sunlight. So these are the component of the light which affect the plant growth and development. And this is about the spectrum, that what are the light are crucial for the plant growth and development. Now come to the photo inhibition process. So in the first slide, we discussed that what is photo inhibition. So highlight, due to highlight, there is reduction in photosynthetic rate known as photo inhibition. So when there is a highlight, it inactivates or damages the PS2. So friend, you know that photosynthetic PS2, this is known as PS2 complex. Okay, so it inactivated or damaged the PS2 complex. So based on this also, there are two types of photo inhibition, that is known as, first is the dynamic and second one is the chronic photo inhibition. So this dynamic photo inhibition generally occurs in the moderate light conditions, and this chronic occurs in the highlight situations. Okay, so in moderate case, quantum yield is decreased, but the maximum photosynthetic rate is unchanged. Means total photosynthetic yield is also total photosynthetic yield reduced, but the PS maximum are unchanged. But in the case of chronic photoinhibition, both photosynthesis and the photosynthesis maximum are decreased. And this is a temporary process, means when there is a highlight, there is a dynamic photoinhibition, but after the low means recovering phase, they will recover, means this is a temporary process. But in the case of photoinhibition, there is a damage of D1 proteins, which is engaged in the PS2 complex. And when there is a damage in the D1 proteins, this is a long lasting. Okay, so by the photoprotective heat dissipation, we can manage dynamic photoinhibition. But in this case, there is no, this is irreversible. Okay, but this is a reversible. Okay, here there is a no photoprotection mechanism in the plant. If there is a damage to the deviant protein, then there will be a decrease in the photosynthetic rate. So friend, this is about the photo inhibition. We will also discuss about some photoprotective heat dissipation, means how like NPQ, that is known as non-photochemical quenching. These are the mechanisms which increase or heat, which are responsible for the heat dissipation. Okay, and friend, this is a light signaling for the access or highlights. So friends, you know that when there is a access light, it is first sensed by the sensing. There is a sensing mechanism, means some photoreceptors already discussed, which absorb the light. Okay, after absorption, there is a change in the thylakoid lumen pH and the change in the redox status. Okay, so friends, you know that there is a proton motive force. Okay, so this force is mediated by the proton concentration. But if there is a change in the, due to excess light, there is a change in the proton concentration and change in the thylakoid lumens. It also change the redox status, means change in the ion status. And also we discuss in the oxidative stress that there is a production of ROS or singlet oxygen during the photosynthesis, and accumulation of chlorophyll intermediates like excited chlorophylls, they are accumulated. So when there is a highlight sensed by the plant, activation of photoreceptors, change in the thylakoid lumens, change in the redox status, means accumulation of glutathiones, production of ROS, accumulation of chlorophyll intermediates. So these changes occurs in the plant. Then there is a signal transduction for the high light stress. Then plant start the acclimatization response. For this, there are some process like chloroplast, avoidance. Then NPQ, that is known as non-photochemical quenching. Okay, also change in the gene expressions and systematic aqua acclimatization. So these are the mechanisms by which plant adopt or acclimatize to the excess light. Okay, so this is clear that what is photoinhibition and what changes occurs during the photoinhibition. So friends, you know that light, how the light are absorbed in this planet or earth or by the plant. So if there is a total 100% of light is coming, this is known as light budget. That how this light utilized by the plant, means total of 100% is light coming and 50% of light is not absorbed. only 50% light is absorbed, and also this 50%, 15% is light is reflected or the transmitted, means not absorbed, and 10% of light is used for the heat dissipation, means as remove as a heat dissipation. Then 20% used for the metabolic, other metabolic process. Only 5% light it utilized for the carbohydrate synthesis or the photosynthetic process. So this is a light budget, that how much light is utilized by the plant. Also friends, this is the leaf anatomy. You know that leaf have first is the epidermis, then mesodermis, then the endodermis. So epidermis leaf have some cuticle surface, leaf hairs. So there are some structures are present on the epidermis. Then there is a mesophyll cells. So mesophyll cells is there. So this is the leaf anatomy. Okay, and this is a C3 plant and C4 plant. So you know, you can see that bundle C cells is present, which is green in nature, or chloroplast are present in the bundle C cells of the C4 cycles. Okay, but in case of C3 plant bundle C, there is no chlorophyll. And this epidermis, this is the epidermis. Okay, this is transparent to the visual light. Okay, so light is coming to the first strike on the epidermis, and this layer is the transparent for the visual light, and this layer work as a convex lens. Okay, so it increase the light to the chloroplast. So this is the first mechanism which increase the light utilization for the plant. Okay, now come to the palisade cells. So mesophyll cells, there is three layers and parallel pillar column. So you know that in mesophyll cells, some are parallelly aligned and some are vertically arranged. Okay, so there is a there is a air space between these, so they improve the light penetrations. Okay, so this is also one of the mechanism to improve the light efficiency. Now come to the other points, like movement of light toward the. So friend, for these there are two mechanisms. First is known as light channeling and second known as sieve effect. Okay, so what is sieve effect? तो sieve का मतलब this chloroplast made like a sieve, means there is a uneven distribution of chlorophyll in the chloroplast. So like this is a chloroplast, and these chlorophyll molecules are randomly distributed or unevenly distributed, which creates the gaps, and these gaps work as a sieve. Okay, these work as a sieve. This reduces the light absorption. So light passes to the other surface of the leaf. Okay. So this is the mechanism which is used for the high light stress. Okay. Next is the light channeling. तो इसमें क्या हुआ है, ओनली सीव इफेक्ट मीन्स जो आपके क्लोरोफिल मोलिक्यूल्स है क्लोरोप्लास्ट में वो अनइवनली डिस्ट्रीब्यूट्स है, सो दे क्रिएट द गैप एंड दिस गैप वर्क एज अ सीव, so light can passes from this sieves to the other surface. Second is the light channeling. So when the light passes to the central vacuoles of the palisade cells, already discussed in the last slides, and spongy mesophyll cells. So there are spongy mesophyll cells and the larger air space are present. Okay, so in the these larger air space are present. Okay, so when light strike on these, central vacuoles and the air these, so it reflect the light. So when light strike on the central vacuoles, so light reflected and there is a light scattering. Okay, so there will be a scattering of the light. So this scattering increase the लाइट पाथवे. ओके, जैसे आपके यहाँ पर बड़े बड़े सेंट्रल वैक्यूल्स है, तो लाइट जब आपका अंदर आएगा तो वो स्ट्राइक करेगा, तो स्ट्राइक करके उसका जो पाथवे है, रेंडमाइजेशन पाथवे है, वो इंक्रीज हो जाएगा. उससे क्या हो जाएगा, आपका जो एब्सॉर्प्शन कैपेसिटी है दैट इज इंक्रीज. सो इट इंक्रीज द लाइट एब्जॉर्प्शन. सो दिस स्ट्रेटजी यूज अंडर द लो लाइट कंडीशंस. ओके, so friends, there are some other mechanism for this light stress like leaf hairs. Okay, तो लीफ हेयर्स क्या करते हैं, जो आपकी लाइट है उसको रिफ्लेक्ट करने का काम करते हैं. ओके, देन साल्ट ग्लैंड्स. सो साल्ट ग्लैंड इमिटेट सम salty water. So this is also reflect the light. Okay, and wax. So presence of wax make the smooth surface. Okay, so it is also increase the reflection of the lights. So they are the some morphological or anatomical changes, anatomicals on the leaf epidermal surface, and these are act as a first line of defense against the highlight stress like presence of leaf hairs, salt glands and the waxy layer. So it decreases the light absorption under highlight conditions. Okay, now other strategy is the solar tracking. Okay, तो ट्रैकिंग से पता चल रहा है कि जो आपका लीफ है वो आपको सन को ट्रैक करेगी. तो दिस लीफ आर मूव अकॉर्डिंग टू द सन फॉर इंक्रीज द लाइट एब्जॉर्प्शन. मीन्स वो अपना पूरा फेस है, मैक्सिमम टाइम आपके सन पर फोकस करेगा जिससे लाइट एब्जॉर्प्शन मैक्सिमम हो. ओके, सो दिस इज ब्लू लाइट मीडिएटेड रेस्पॉन्स एंड एडजस्टमेंट ऑफ लीफ ब्लेड. सो उसमें जो आपका लीफ ब्लेड है वो आपका चेंज होता रहता है अकॉर्डिंग टू द सन पोजिशंस. ओके, दिस इज नोन एज सम मोटर सेल्स आर रेस्पॉन्सिबल फॉर द सोलर ट्रैकिंग. ओके, विच इज प्रेजेंट इन द लीफ ब्लेड and vertical position at sunrise and eastern direction. ओके, तो आपका जैसे सन राइट जैसे ईस्ट में होगा तो वो ईस्ट में होंगी, और जैसे ही आपका नाइट आएगा वो हॉरिजॉन्टल हो जाएगी. ठीक है, दीज लीव्स मूव विद सन प्लस माइनस 15 डिग्री. जो आपका सन है उसके एंगल से 15 डिग्री मूव करती है. दिस solar tracking are found in the some plants like lupines. Okay, also cotton, soybean. Okay, so these plant solar tracking. So this is one of the strategy to increase the light absorption. Then, so friends, there is a one mechanism that is known as sun flags. Okay, so these are the patches of sunlight. So friends, you know that if there is a dense canopy and there is some vegetation on the lower surface or the soil surface, then there is some patches or some places on the forest which are low dense, and they make the high light. So highlight penetration can occur in these slides. So these make the plates on the earth's surface. Okay, so here only short period of cadence occurs in which some shady plants absorb light in this time and made food for the whole day. Okay, इसमें क्या है कि आपका जैसे फॉरेस्ट कंडीशन है तो वहां पर क्या है आपकी डेंस कैनोपी है, लेकिन कई कई प्लेस पे क्या है कि जो डेंसिटी है वो कम है. तो जब आपका सन उस पोजीशन पे या एंगल पे आएगा तो वहां पर आपका ज्यादा लाइट अर्थ पर पहुंचेगा. तो अर्थ वेजिटेशन पे ऐसे क्या होगा, आपके सन फ्लैक्स और पैचेस बन जाएंगे. तो at that time these canopy accumulate the or absorb more light and start photosynthesis or more photosynthesis. Also there is a movement of leaf that is known as heliotropin. Okay, so diheliotropin and the para-heliotropin. So this is a vertical movement, leaf angle. So for the maximum light absorption, and para-helio means horizontal leaves, so reduce the absorption of light. Okay, so these are the movements of the leaves for the increase or decrease the absorption. So friend, these are the some changes for the light conditions. Now also some acclimatization response, already discussed that chloroplast avoidance. So what happen in these conditions? So in this condition chloroplast move to the side, position parallel to the incidence light. Means आपकी जैसे ये लाइट आ रही है तो आपके क्लोरोप्लास्ट इधर एक्युमुलेट हो जाएंगे. ओके, टू रिड्यूस द light absorption. And these movement mediated by the phototropin and the cytoskeleton, that is actin and the calcium. Okay, so this is also one of the acclimation response for the highlight stress. And this is also another mechanism that is known as photophobic movement in chlamydomonas. So this exhibit the flagellate mobility controlled by the membrane depolarization and activation of voltage gated calcium channels. Okay, so there is a movement of flagellate, means photophobic, opposite to the light. Okay, and flagellates help to the this photophobic movement, and membrane depolarization, activation of calcium channels. And rhodopsin A, V, that is known as CSR A and B, these control the phototaxis movement. Okay, also these sun plant and the sun loving plant and the shade loving plants have some anatomical or some biochemical features, like these shade leaves increase. क्योंकि जो आपका shade plant है उसमें तो क्या होना चाहिए कि हम how we can increase the light absorption, and when there is a or sun plant में क्या होना चाहिए, how we can avoid the light. Okay, तो ये एक दूसरे के अपोज मैकेनिज्म चलती है. तो shade leaves increase the light capture by the high chlorophyll. एक तो टोटल क्लोरोफिल आपका ज्यादा हो जाएगा और क्लोरोफिल भी इंक्रीज कर लेंगे, जिससे कि आपका लाइट का कैप्चराइजेशन ज्यादा हो सके. आल्सो देयर इज ए थिनर लैमेली, जो आपकी लीफ सरफेस है जो उसको स्लो डाउन कर, जो आपकी लेयर्स है जो मेसोफिल्स की वो रिड्यूस कर लेते हैं, जिससे क्या होगा आपका लाइट ज्यादा एब्जॉर्ब होगा. आल्सो दे हैव हाई PS second to the PS first. Okay, because you know that PS second is the light levels, but shade plants में ये आपका ज्यादा होगा. Okay, also there is a more antenna chlorophyll. इसके अपोज में सन लीव्स में क्या होगा, they have large xanthophyll cycles. Okay, क्योंकि वहां पर हमें we need means we need photoprotection mechanism. Okay, तो xanthophyll cycles क्या करता है, आपका हीट डेसिपिएट करता है. देन थिकर लीव्स ज्यादा होगा, जिससे कि आपका कम लाइट आपका क्लोरोफिल तक पहुंचेगा. एंड लार्ज पैलिसेड सेल्स मींस आपका लार्ज डेंस सेल्स होगा. ओके, जिससे आपका लाइट एब्जॉर्प्शन नीचे कम पहुंचे. ठीक, सो दीज आर द सम मैकेनिज्म. आल्सो नॉन फोटोकेमिकल क्वेंचिंग. सो व्हाट इज नॉन फोटोकेमिकल क्वेंचिंग? तो quenching of chlorophyll fluorescence by other than photochemistry. ओके, तो जो आपका फोटोकेमिस्ट्री में जब आपका एक्साइटेड इलेक्ट्रॉन जाएगा तो यहां से कुछ तो आपका फोटोकेमिस्ट्री में जाएगा. ओके, और कुछ आपका नहीं जाएगा, तो वो उसमें क्या होगा, हीट के रूप में या फिर फ्लूरेसेंस की फॉर्म में यहां पर होगा. एंड दैट इज रेस्पॉन्सिबल फॉर द ऑक्सीडेटिव स्ट्रेस. So xanthophyll cycle work as a volume knob, means controlling this fluorescence. Okay. So NPQ is the non-photochemical quenching is the strategy to reduce the fluorescence. Okay. Then leak movement already we discussed about this dihalotropin and the parahalotropin. These are the movement to reduce the or increase the light absorption. Okay. Also there is a photochemical quenching, means quenching of the excess heat during the photosynthesis process. Means there is a change in like a decrease in the antenna size. Okay. Like, and in this LHCb have important role. Okay, so when there will be a decrease in the antenna size, they dissipate the heat. Then there is a some thylakoid associate kinase like STN7 and H. These phosphorylate the LSCB. So these are also important role in the quenching of the fluorescence. Okay, means these are the some strategy used against the highlight stress. Okay, increase in the STN7 and 8. So it will increase the phosphorylation of LSCB. Okay, will decrease the antenna size. Also, there is some transcription factors like J812, APX1, that is antioxidant, HSF, heat shock factor, HSFA, HSF5. So these are the some transcription factor which also have important role in the mitigation of this highlight stress or acclimatization to the highlight stress. Also, development of fluorescence mutant show that there is no ROS production. So we can say that these fluorescents are responsible for the production of reactive oxygen species in the plant during photochemistry process. Also, this is xanthophyll cycle. Okay, so friend, this is this yellow one is the light intensity. Okay, so you know that during morning time there is low light, and during afternoon there is highlight, and again at evening there is low light. Okay, so this is common feature, and this is a molecules, and this is known as bioxanthin. So when there is a low light, the concentration of bioxanthin is highest. Okay, but when there is a highlight, there is low bioxanthin. But opposite to this zeaxanthin and anthraxanthin is high under the highlight condition and low under the low light condition. So we can say that under highlight condition, this bioxanthin change into the zeaxanthin and the anthraxanthin. And low light condition, this zeaxanthin is again converted to the bioxanthin. And epoxide and deepoxide enzyme converted to this bioxanthin and zeaxanthin. So friend, this is about the mechanism which have important role during the mitigation of highlight stress. So I hope you understand today's lecture that how this highlights affect the plant growth and the photochemistry, then how plant acclimatize this challenging environment or fluctuating situation. So thank you very much.